Hello, dear students. We will continue our next lecture, lecture number 5. Last time we have seen about cell organelles plasma membrane. Now we will see the rough and smooth endoplasmic reticulum that we have discussed at last. Then, in spite of that, plasma membrane and endoplasmic reticulum, the remaining cell organelles are Golgi bodies, lysosomes, mitochondria, ribosomes, etc. So we will discuss one by one. So Golgi bodies, these are also called as Golgi complex because these are many in number, and as they are identified by the scientist Golgi, the name is given Golgi complex. They have cup or disc shaped structures attached to the endoplasmic reticulum on one side. It is packaging and forwarding center for many secretory products, or we can say the secretory molecules materials are being produced here in endoplasmic reticulum, which later on form the Golgi bodies. It is believed that the vesicles of Golgi bodies are associated with lysosomes formations. Means all the cell organelles are formed by the union of different tissues molecules, and lysosomes are the final products from the endoplasmic reticulum. So these are the lysosomes. Golgi bodies are the secretory product forming cells, or we can say secretory product forming material, and lysosomes are also called as suicide bags. Here is the word suicide bag because they throw its secretion directly on the food material inside the cell. So these are many in number. Lysosomes are tremendously found in the cells and freely distributed all over the cytoplasm, and these contains about 50 different types of hydrolytic enzymes. Lysosomes are very minute but have 50 different types of hydrolytic enzymes and acid phosphatase groups. Functionally, lysosome concerned with intracellular digestion of the food. Lysosomes play key role in degeneration of the tail during exactly the metamorphosis in frog. Due to their autolysis nature, means cell breakdown nature, autolysis means cell breakdown, they are termed as suicide bags of the cells, or they throw their secretion directly on the food material or to the material wherever it requires degradation or digestion, totally digestion. Fourth organelle is the mitochondria, to which we generally call as powerhouse of the cells because they generate adenosine triphosphate, ATP generator, and they are concerning with the generation of energy rich compound ATP through its electron transport chain or electron transport mechanism. This is a peculiar mechanism. This is the specificity of mitochondria, electron transport chain and oxidative phosphorylation. So these are the key factors which are performing the physiology of mitochondria. The number of mitochondria in a cell is variable according to the functional status or to the physiology of the cell, and the shapes are also variable, but generally they are of spherical shape, or mostly we say that mitochondria are spherically or rod shaped. It has a double membrane, encloses its own DNA ring and 70S type of ribosomes. So ribosomes are two types, 70S and 80S. So they are 70S type and the enzymes of the respiration. or we can say respiratory enzymes plays a key role in electron transport mechanism and forming the ATP. This is the main function of mitochondria. These are also randomly scattered in the cell. Ribosomes. These are very small granular units and they take part mainly in protein synthesis, or we can call it as, or we can term it as factories of protein synthesis, because they are also in tremendous number and they help in formation of protein molecule by translation and transcription mechanism. So ribosomes are freely distributed in cytoplasm and attached to endoplasmic reticulum and outer nuclear membrane. These are very minute. These are non-membranous particles and made up of two subunits. If they are 70S, it is 50 plus 30 subunit. Then ribosomes are sites where proteins are synthesized. Ribosomes are the main site where proteins are synthesized, and therefore these are also called as factories of protein synthesis, because they produce tremendous number of protein molecules by the process of protein synthesis, and which goes through the central dogma process that we will see in detail in the further topics. So next one, sixth organelle is the centrosomes. These are found located near the nucleus. Centrosome consists of pair of centrioles and plays a role in formation of spindle fiber or spindle apparatus and basal bodies for cilia and flagella. Or we can say the spindle formation we have already studied in mitosis and meiosis. The chromosomes are separated by using these spindles and these are formed by the centrosomes. Seventh cell organelle is vacuoles. So these are primarily found in few lower animals. They are bounded by single selectively permeable membrane called as tonoplast. They maintain turgor of cells. Contractile vacuole in animal are concerned with the osmoregulation, and the vacuoles generally formed by the cell organelles in cells as ribosomes or lysosomes or RER or Golgi bodies. These all are related with each other, and endoplasmic reticulum is responsible for the formation of different other organelles. 8 is microtubules and microfilaments. The cytoplasm is generally traversed by numerous ultrafine tubules of a tubulin protein. This is the tubulin protein called as microtubules. And the function of these microtubules is the transportation of water, transportation of ions or small molecules, and formation of spindle fibers of the mitotic and meiotic cell division. Whatever the type may be, whatever the method of cell division may be, but the spindle fibers are formed by using these microfilaments or microtubules. The cytoplasm of most of these animal cells contains ultra-fine proteinous solid material, and that ultra-fine proteinous solid material is called as microfilaments. They maintain the structure of cell and forms contractile components of muscle cells. So in this way, the microtubules or microfilaments are responsible for the formation of molecular or fibrous material to give the strength to the cells. Cilia and flagella, these are the another organelles. The cells of many unicellular organisms and ciliated epithelium of multicellular organism consists of some hair-like cilia or cytoplasmic projections outside the surface of the cell, and these are generally called as cilia or flagella. These are the two outgrowths of the cell, or we can say organelles. They help in locomotion of the cell. It consists of 9 outer fibrils around 2 large central fibrils. As the roots are there, taproot is the main root and other fibrils are the another. Likewise, the 9 outer fibrils around the 2 large central fibrils. So in this way, the cell organelles are studied one by one. Then these are about the cytoplasmic organelles. Now we will see the nucleus, the central part in which nucleolus is there. The nucleus is the central body of each cell, particularly eukaryotic cell, and controls the activities of the cell. Nucleus also located in the center of the spherical cells and controls all vital activities of the cytoplasm and carries the hereditary material, that is DNA, in it. Typically, nucleus consists of nuclear membrane, then nucleolus at the center, then nucleoplasm, and chromatin network, that is chromosomes or DNA and RNA. So these all are there present in the nucleus. Outer nuclear membrane is connected with endoplasmic reticulum, whereas inner connected with chromatin fibers. The interrupted region is called as nuclear pore, through which mRNA passes out during transcription and DNA for protein synthesis. So this is the interrupted region where the exchange of the nuclear material is one by one carried out. Next one is the structure of plasma membrane. So this is all about, we have seen cell organelles. Now in detail in syllabus, the second point is structure of plasma membrane. The plasma membrane is also called as cytoplasmic membrane, or we can say the membrane which encircle or around binds the all cytoplasmic matrix is called as plasma membrane or cell membrane or plasma lemma. The term cell membrane was coined by C. Nageli and C. Kramer in 1855, and the term plasma lemma have been given by J. Q. Plovy in 1931. It encloses every type of cell, both prokaryotic and eukaryotic cells. It physically separates the cytoplasm from the surrounding cellular environment. Plasma membrane is ultra thin, means very thin, elastic, then living, dynamic, and selective transport barriers. It is a fluid mosaic assembly of the molecules of lipid or phospholipids, proteins, and carbohydrates. So they are just not binding but also supporting, also surrounding, and barriers to the selective transport material throughout the membrane. Then in that structure of plasma membrane, the two models are very famous. The bilayer model of Danieli and Dawson, this pair have identified that there is a plasma membrane which have double membranes, therefore it is called as bilayer membrane or sandwich membrane. It is on the basis of the comparative data which are available regarding the surface tensions of the lipid layers and cell surface, then collected by H. Davison during early 1930s and J. Danelli during 1935. So within that 5 years of period, these both scientists have identified different model and their organelles. So different working mechanisms. So these both have concluded that the plasma membrane contains a good amount of proteins in addition to the lipid layer or lipid bilayer layer. They hypothesized that there are different things about the membrane. So this is the layer, we can see sandwich model of Danieli-Davzeni, and this is pore structure in the model of Danieli-Davzeni. This is a polar pore structure. Pore means opening. So this is the membrane, how the opening is formed in the membrane. First is that the polar or hydrophilic membrane heads of inner lipid layer are embedded in the surface of the aqueous cytosol, whereas the heads of outer lipid layer are embedded in the surface of aqueous extracellular medium. In this way, so these are the heads of the lipid molecules. This is the lipid molecule file. This is also lipid molecular file inside, and outside these are the protein molecules. So inside lipid and outside protein, therefore we can call it as a bilayer model. So these are the strands to which called as lipid bilayer, or we can say lipid layer in simple. This is protein layer. So this is the head of the lipid molecule and this is the inner rod, we can say strand. Second, the non-polar or hydrophobic, this is made of fatty acid molecules and the tails of lipid molecules of both layers, and they are directed away from the respective aqueous phases. And hence, these faces each other and forms a middle core region of the plasma membrane. Then third one is the polar heads. These are the polar pore, these are the polar groups, these are the protein material small, these are the non-polar groups, this is the lipid inner lipid bilayer model. These are the two different models but are shown picture as a complete understanding that this is the pore. The polar head of both lipid layers are coated or covered by hydrophilic proteins which form almost continuous layers. So this is the picture in which the sandwich model or bilayer model of Danieli-Davzeni is explained, and here the pore structure in model of Danieli-Davzeni, how the pore is formed, here we cannot explain very well, but in this way we can explain the pore in between these two. This is the very magnified part, this is less magnified part. So this is the pore that forms the inner gap in between these two membranes. If it is naturally, no problem. If it is made, it is quite difficult. So these are the larger molecules, hydrated proteins covering outside, then followed by the head of the lipid molecules, then followed by the tail or strand of the molecules. In this way, this is one side, and another side is at the inner cell or wherever the animal touches itself. This is the inner side, or we can say cytosol. This is extracellular medium, and this is cytosol. Cytosol means cytoplasm, we can say simply. So unit membrane model, only two topics we have to study here. First we have seen Danieli-Davzeni model, and second is the unit membrane model of Robertson. In this way, in this model, the plasma membrane was actually observed for the first time with the help of electron microscope during the early period of 1950. It was found to be a three-layered, we can say trilaminar structure showing two dark staining lines separated by a middle light staining line in a dark railway track pattern. We can say where as the railway track pattern is there. So sandwich model and likewise the unit membrane model is arranged in the cell. So each dark line is represented or represents a continuous sheet of mostly small and globular hydrophilic proteins together with the underlying polar heads of lipids molecule. So it depends on the working of the particular molecule. Whereas the middle light line represents a bilayer of a non-polar fatty acid tails. Bipolars might be the sunlight, might be the focus is not there reaching, so that this may show as such activity, whereas middle light lines represent a bilayer of non-polar fatty acid tails of lipid molecule of both layers. So it was also found that the proteins of external and internal layers are dissimilar. These are not similar. Even though the molecular arrangement is same, but structure is not same. It varies according to the animal or according to the organ. And so that the trilaminar structure is asymmetrical, means there is no similarity in there. And since certain cell organelles like nucleus, mitochondria, chloroplast, etc. are surrounded by two concentric membranes, and each membrane having this trilaminar structure. J.D. Robertson is the new scientist. He has been studied in 1957 and proposed that the trilaminar structure represents a unit membrane. We generally call it as a unit membrane instead of plasma membrane, because this is not a single one. It is a large membrane and it works with the role of animal body. And this concept confirms that the plasma membrane is a water soluble barrier, so that only lipid soluble substances can pass directly through it. So this is the extracellular medium and this is the intracellular medium. Suppose that, so in that the hydrophilic head of lipid molecules are arranged serially at both sides in the membrane, and the lipid bilayer model is here. So one, and this is two layer. This is lipid bilayer model, or we can call it as this single piece is called as the tail of the lipid molecule. This is the head of the lipid molecule. Very simple that I have seen in the color structure. So in this is the outer protein layer and this is the inner protein layer. In between these two protein layers, these two lipid layers are there. Therefore, it is called as exactly a lipid bilayer model, or we can say lipid bilayer structure of the cell membrane. So this is about the Robertson's unit. So Robertson's unit membrane model is showing that likewise the sandwich model, outer protein materials and inner lipid materials over there on the strands, or we can say a central line. So this is about the extra and intracellular cell membrane, then it's the cell organelles, the structure and function of the organelles, and all this about today's all the membranes and cytoplasm. So in eukaryotic cell, we have seen all about the cell structure, matrix, plasma membrane, cell organelles, and their functions also. So this is about total eukaryotic plasma membrane and cytoplasm. So we will see in further to the next period about the fluid mosaic model. This is also a second type, but we will see in the and next lecture. Till then, see you.